Welcome to The Mind, the Cure Protocol for August 16, 2026. We begin with what changed, not merely what was announced. One movement in this path has already reached care. The rest are research signals that could open the next path, but each still needs proof. We will start close to real people, then trace the evidence back to the targets and questions that matter next. This protocol is a living loop. People bring the questions that matter. Direct evidence changes the map. The source ledger remembers what we have already seen and checks the original date before calling it new. Every signal ends with one practical next action, the experiment, validation, collaboration, or care decision that could move it closer to real benefit. Researchers reported in NPJ Aging that the FDA-approved lipid drug Gemfibrozil extends healthy lifespan across evolutionarily diverse species by inhibiting the dipeptide transporter PEPT1 SLC15A1, rather than its canonical PPAR target. The result matters because it reframes aging and disease modulation as an engineering problem in nutrient uptake control. TAA-guided biology can increasingly identify lever points in conserved metabolic circuits, turning existing molecules into precision interventions. For people within the stated indication, this is now a regulated care option. Treatment decisions still belong with patients and clinicians. Approval establishes regulatory status for the stated indication, not proof of superiority or the right choice for every person. Next, watch real-world uptake, safety, and outcomes within the approved indication. A Nature Communications Phase II study by Mo Cheng and colleagues tested Sintilimab plus the Bevacizumab biosimilar IBI305 with chemotherapy as first-line treatment for advanced gastric or gastroesophageal junction adenocarcinoma, establishing a clinically actionable combination in a hard-to-treat solid tumor. If confirmed, this could help clinicians see disease signals more clearly and focus the next decision. The useful part is precision, not magic. A clever sensor still has to prove it helps a real clinic make a better decision. Next, show in independent studies that the measurement improves a real clinical decision. Researchers led by Arsen Aqib, Hamid Ravi, and colleagues introduced SynCellNet in Scientific Reports, a generative AI framework for synthesizing single-cell RNA-seq expression profiles and validating that the synthetic cells are statistically similar to real data. The result matters because single-cell transcriptomics is a bottleneck for turning disease biology into computable structure. By expanding scarce and imbalanced datasets, SynCellNet pushes biomedical AI toward the intelligence inversion in which algorithms learn the cellular state space well enough to make disease less an observational mystery and more an engineering problem. If this holds up, it could give researchers a clearer target for the next experiment, validation step, or collaboration. The useful part is a clearer target, not a finished solution. Independent evidence still has to do the hard work. The next question is whether independent evidence confirms the finding and shows a responsible path toward real-world use. The practical read is simple. These are signals, not promises. But each one is a concrete target that researchers, clinicians, engineers, funders, and the people most affected can connect to a next step. Keeping the evidence visible is how the cure protocol stays current without pretending the work is finished.